Hello friends, welcome to BMH Learning. This video is about metagenome assembled genomes. A metagenome assembled genome is a single taxon assembly that is based on one or more binned metagenomes. MAG submissions are submitted at the same level as isolate genomes and are distributed within INSDC in the same way. As an environmental sample can contain many duplicate genomes of the same organism, and as MAG assemblies are more prone to contamination, it's requested only the highest quality unique taxon submissions are submitted as MAGs. There should only be one MAG submitted for each species within a biome. The quality of a MAG is highly dependent on the species, genome size, its abundance in the community, and the depth at which it's sequenced. Two important things that can be measured to know quality of MAG are completeness, that is the MAG a complete genome? And if it is contaminated, that is, does the MAG contain only one genome? This was all. Thanks for watching.